Good day, everyone, and welcome to our Drug Discovery webinar. My name is Emma, and on behalf of Oxford Global, I'd like to thank you all for joining us today. Today, we have Lilly Zhu, Senior Vice President, DMPK and Exploratory Toxicology Department at ChemPartner, who will be presenting on the application of in vitro and in vivo ADME, PK, and exploratory toxicology assays to support lead and candidate selections. After the presentation, we will have a 5-minute Q&A session. Please submit your questions through the chat option on the bottom of your menu display. The full recording of the webinar will be made available to download on our secured website, and the details will be sent to you after the webinar. Without any more delays, I'd like to get started with Lilly's presentation. Lilly, thank you so much for joining us, and over to you.
Good morning, good afternoon. Thank you for listening to my webinar. And again, today's topic is application of in vitro and in vivo ADME, PK, exploratory tox assays to support lead candidate selection.
Thank you so much, Lilly. Would you mind making your slides full screen?
Full screen. Am I
That's
right?
just the button to the left of the minus. If you go down to the bottom of your screen, there's a plus and a minus, and there's a button to the left of the minus on the red part.
Oh, okay. Yes. Got it.
Thank you.
So, so I want to introduce my company first. ChemPartner on our DMPK and Tox service coverage, drug discovery, preclinical, and IND filing services. And as you know, for the drug discovery early stage, we usually run target identification, validation, lead generation, and lead optimization and disease screening. And in the preclinical stage, we run bioanalytical studies, DMPK studies, and safety and toxicology study, besides in vitro and in vivo efficacy studies in the pharmacology area. And for the IND filings, we will do in vitro ADME studies and in vivo ADME studies. And these are our expertise in this area. We help lots of companies, US, European, and Chinese company to achieve this during our service work. And as you know, how do we integrate ADME assays? ADME means toxicology too. Assays in drug discovery. We have different stages, in terms of the compounds. For example, lead generation, lead optimization, lead characterization. At the lead generations, usually you have a lot of series of compounds. In each series, you have 25 to 50 compounds. And for the biochemical and the cell-based assays, you usually look at the potency, your target. For DMPK part, as you see in the highlighted red markers, is we can run rat and human microsome stability assays. We just want to look at the percentage of the remaining. This is a very early stage because lots of compounds there. We only use like a high throughput screening assays to generate some data or rank order. And also we run CYP3A4 2D6 inhibition. We look at the percentage inhibition. And then we do some plasma protein binding. We want to see, how tight your compounds bind to the plasma binding, because later on you will use the data to design dose for efficacy dosage, because the free fraction is the driving force in terms of efficacy. Then we do some log D solubility assays. We use the kinetics method. And some of the companies, they want to do some early PK studies, so we run that too. Then when the series move forward, we go to the lead optimization. You have a good potency in terms of cellular, sometimes in vivo models you kick in now. Then you will choose 2 or 3 series, which is have good potency. And then start more detailed ADME assays like log D solubility. Usually we use thermal, or also we do chemical stability, microsomal hepatocyte stabilities. At that stage, you still use human or rat, or maybe more species, are relevant to your PD assays. But we are generate intrinsic clearance, T-half, all these data for the stability. Then we do CYP inhibition, also induction kick-in. Maybe we want to use one donors, like human donors, see if, has any induction issue, DDI issue. And we do some TDI, time-dependent inhibition shift assay too. Certainly, we have previous some PK data, or at this stage, we run some PK studies. And when you see the low bioavailability, you were looking at the KCO2, transporter related issues. Certainly, plasma protein binding, you will look at the relevant species in terms of your PD. And we do some MET ID because we want to fix some of the liability in terms of unstable compounds in microsomal hepatocytes. At this stage, in the lead optimization stage, we also run some hERG assay. Well, we want to root out potential cardiovascular toxicity. So then we want to do move forward again, lead characterization. Again, these is the assays. You want to these are the highlight assays you want to do. And also we want to do some phenotyping assay, which is required by FDA. And the TDI, such as we want to do some KIK-NET. If you have some problem in terms of time-dependent inhibition, you can combine with the hepatocyte induction assay, or looking at mRNA, or looking at activity to see if you have a TDI issue. Also, you can generate K small I and K big I's, and these are the for the simulation later, or maybe this stage, or when you go to the candidate. And then certainly we do some in vitro or in vivo toxicology assay like AIMS or MNT. So I want to do a little bit more detail in terms of metabolism assay. When I mentioned that before in different stages, as I said, metabolism assay, we have microsome stability, mainly for phase 1 metabolism. Then we do hepatocyte stability because when you do some, when we run microsome stability and also PK studies, sometimes you see microsome is very stable, but your PK, intrinsic clearance, it's really fast. So there's a disconnection, IVIVC disconnection. So we might think, hepatocytes can be a tool because hepatocytes not only combine phase 1, but also phase 2 enzymes. Certainly, at this stage, we'll also do some stability in different matrix, studies, and also reaction phenotyping, also AO stability studies. This is the studies we run about a week. You can have all the data. And then we do the DDI, second, as I mentioned, CYP inhibition, time-dependent inhibition, CYP induction. These are all the early stage assay. And also we do some KI next. This is more late optimization stage assays. In the transporter inhibition, at this stage you can use these type of assays to see potential DDI. And again, I mentioned protein binding assays, which is important. Lots of people use free fraction and to calculate, for the dosage later on for the for PD assays. And some of the clients want to do blood to plasma ratio, and these assays we can run like within a week. And certainly, as you know, there's equilibrium dialysis and ultracentrifugation protein binding assays. These two assays has pro and con. Equilibrium dialysis assays, very quick and rank order wise, you can use this. Ultracentrifugation, some of the clients want to use for more detailed, but still it has the sort of non-specific binding, all these, in terms of middle layer, when you ultracentrifuge your plasma and protein bindings, so it's not absolutely which we choose, but we need to choose carefully how to interpret the data, which is very important for us. These kind of studies at turnaround time, a week, you can get it done. And the transporter assay, as I mentioned, data, when you see low bioavailability, you're wondering if if a transporter involved in terms of efflux, in terms of low bioavailability. So you typical we use like KICO2 for transporter assays. We do quick turnaround time, so 1 week, because we have lots of KICO2 assay requests. So we use the cell line culture is continuously, and also we for help CNS or some projects we do MDCK, MDR1, MDCK, BCRP, or MDCK, MDR1, BCRP, double transfected. And these type of assays for us, we can generate the data 1 week. So we can sort of solve some issues in terms of transporter. And then we have physical chemical property assays. We do have how to measure that solubility, as I mentioned, thermal and kinetic, sort of a solubility. We do log D, chemical physical stability, and a particle size assays. Turnaround times are like 1 week. And the next assay is more advanced assay, CYP phenotyping assays. We do these assays according to FDA guidance, measure different isoforms. This is a part of FDA submission. I want to mention that the assays run in ChemPartner's more detailed assays are sort of compliant with the FDA guidance, and also with the big pharmas, assays. I came from Amgen and Sanofi, so when I look at the detailed assays, content, the way we run, it's similar to their assays. So the phenotyping assays about 1 week we can generate for you. All these assay results can be part of a submission because, as I mentioned, our assays are compliant with the FDA and guidance. So what we need to do is that if clients say, "Oh, I'm gonna do IND filings," what we do is like put all the results in a IND filing compliance report, and we have lots of company work with us on filing FDA submission, and what we do is like put the report together and send to our clients, and then they put into the IND filing submission. So the other assays we have is the ID assays. As you see, at a different stage, discovery, preclinical, and clinical development, we focus more early discoveries like, soft spot analysis, reaction, METs, ID screenings, support lead optimization. And the slide is the MET ID identification assays. Basically, is like the purpose on the right side. I'm not gonna go into detail. You can look at, and take a look later. You will have the slides, and then you will look at the tools. What's the purpose? How we use the tool. To do MET ID studies. Early stage, as I mentioned, we can do in order to save the resources, particularly for small virtual companies, we can do a shift MET ID assays, just give you some ideas, what's the hotspot. But later on, we can do detailed MET ID, for FDA submission. We can do in vitro and MET ID, all species, as you'll see. And also we do in vivo, rodent, also dog and monkeys, MET IDs in vivo. We do a lots of PK studies. We have the techniques as models. As you see, we have a cannulation, dosing, sampling, CSF collection, and tissue collection, organ perfusion, rodent. We do lots of PK/PD relationship analysis because we have clients working with us in like oncology, CNS, metabolic disease area. We do a lot of work with the pharmacology folks together. We design the dosage for them based on in vitro and in PK data. And also we do a lot of formulation support, for PK and exploratory tox. As you know, the chemists they don't make the perfect compounds. Always, most time make very insoluble compounds. We do have the formulation support for a routine PK and exploratory tox. Particular, we want to have a cohesive approach, prepare clients for GLP tox. So we do need the formulation support for the exploratory tox. So these are the PK models, as you see. These are the summary tables, what we do. And we have different species. We do surgeries like cannulations, dosing. We have a different route. And the sampling, we do have a different places, for sampling. And you can take a look later in terms of more detail of where we do sampling, dosing, and the surgery. Again, I mentioned we do have full capacity of formulation capability. We also work with the CMC because some of the clients want to do GLP tox, further. We do compound management, formulation preparation, formulation screening. We have a different formula. We've been working with so many clients, and different clients have different, criteria. Also one of formulation selection. So we also do formulation analysis. So overall, just, we have very strong formulation capability, and we can do different kinds of formulation for you. And also I want to mention for early stage formulations, kind of a different, like rodents, different from dog and monkeys. Some of the formulation cannot be used in dog and monkeys because due to a toxicity. And this is just an example mouse PK, serial bleeding PK, in various serial bleeding. These are PK studies in order to, reduce the animal numbers. We pay a lot of attention to animal cares, so welfare, and also so reduce the animal number from 70 to 9. And also early stage, we don't, chemists, they don't make enough compounds, only few milligram compounds. So we'll reduce the compound usage and also minimize the PK data variability. We do serial bleeding, in mouse and rodents. And you see these the data we generate. Certainly it is the whole blood data. You need to figure the blood to plasma ratio. Sometimes 1:1, sometimes not really. So we need to, you need to pay attention to convert the numbers ratio in terms of the PK data. The next one is the BDC Monkey biliary, renal, and fecal excretions study. These shows, how capable we are in terms of running all these, not only rodent PK study, but also dog and a monkey. And also we perform lots of surgeries, for different purpose, for PK studies. And the next one is the mouse and the 1 PK profile for biologics. Our company not only, today I'm more focused on small molecule in terms of in vitro ADME and in vivo PK and tox, but we do have a capability in terms of biologics. These just examples how we run for the biologics PK studies. And for in vitro exploratory tox assays, we do hERG assay and NAV 1.5 screening. And I mentioned AIMS test. This is the in vitro genotoxicity assays, AIMS test. We do have full strains, and also we do mini one too. 2 strength for the AIM studies, and the turnaround time is like 4 weeks. And next is the integration of the ADME assays in preclinical studies stage. And this is a more candidates staged assays we are use for our clients. The slides like 1 to 20, this is more like a lead generation, sort of a part before. And this is this slide is more like talking about candidate, how we help clients generate or prepare for candidate or FDA submission. Basically, these are the detailed assays, full PK, microsome hepatocyte stability, CYP inhibition. We do 8 or 9 isoform and IC50 generate, and TDI, KIK, CYP induction. At this stage, FDA requires 3 donors. So we do previously early stage, you can do 1 donor or 2. And this time you need to have full 3 donors. And you do generate EC50 Emacs. All these in vitro data you can be used for simulation. You can look at one of my papers before, like a Journal of Drug Metabolism. We have these kind of a simulation papers. You can take a look how we use these data, particularly CYP induction data, to predict potential drug-drug interaction in human. And then we do the plasma protein binding, which is necessary, and also transporter, not only efflux uptake too, and according to the FDA guidance, MetID, you have microsome hepatocytes. Also we, I can talk about later about the IND filing. So you're going to generate in vivo MetID data too. And this stage, we also start running all these single dose, repeat dose toxicity studies, tolerability toxicity studies. And so for in vivo, we do some in vivo MNT in mouse. At this stage, this is more like a non-GLP, but give you some idea what happened to your compounds. And this is the single dose. These are more detail how we design single dose toxicity studies. Turnaround time 3 weeks. And repeat dose toxicity studies depends on how many days, like 5, 7, 14, 28 days. And these are the content, the table is more detailed. Certainly we do lots of clinical pathology, hematology, serum chemistry, and the urine studies, assays. And tolerability studies, and look at more detail when you get a chance. And turnaround time is 2 weeks. You can generate some data for your toxicity tolerability studies. And this is the clinical pathology content and histopathology we do for lots of clients. And this is the tolerability examples, how we run in the cyno monkeys, not only the TK, but also we look at the clinical pathology, clinical observation, and also histopathology. We look at the skin, eye, and the lung. And these type of studies, we can run routinely for our clients. And the last is the IND filing package. As I mentioned, we do a lot of IND filing package for our clients, studies for our clients. When you take a look of this IND filing package, you will see that in vitro part, we do absorption, distribution, KCO2, plasma protein binding, transporter, metabolism, clearance. We do liver, microsome, hepatocyte, stability, CYP phenotyping, ID, and we do in microsome and hepatocytes, and DDI, we do CYP inhibition, CYP induction. And for in vivo part is the bioanalysis. I didn't focus much on the methods, feasibility, and the validation earlier, but these are the part that are really important. You want to do all the methods of validation, different metrics. And also for the PK part, we do single dose PK study in rodent and the non-rodents, or you were related, you will choose which species are more related to human based on the, MET-ID in vivo data. And also we do 7 days repeated dose PK studies in rodent and non-rodent, and a tissue distribution excretion test in rodents. These are all, these are the studies you can choose do or not to do. And then the last part of toxicology package is a formulation bioanalysis, same method, feasibility and validation, safety pharmacology, hERG assay, CNS assessment in rat, and cardiovascular and respiratory studies in dog, and also the general toxicity, single and the repeat dose in rodent or and non-rodents, and genetic toxicity aims MT, also chromosomal variation studies, and certainly the development toxicity. These studies more specific for each clients. So basically, the studies I mentioned, we run all these studies, for our clients and generate all these data, to support eventually the client's IND filings. And this is my address, email address. If anything, you have any questions, don't hesitate, contact me directly. And we definitely can help your companies and you to solve some of the issues or running assays. And thank you very much for listening to my talk.
Thank you so much, Lily, for a fantastic presentation. Now, I would like to open the floor for discussion. Remember to send us your questions using the chat feature of the platform. So the first question that we have here, Lily, is, are those assays also adapted for compounds such as PROTACs, or do they need to be somehow adapted?
PROTAC is the kind of a new things for us. We are learning it, but we have the expertise from other big pharmas just joined us. We can work on this, with you and gain more experiences in that area.
Fantastic, that's great, thank you. Another question we have here is, what kind of human renal cell lines are you currently using? And do you have any experience with the CIPTEC cell lines?
This is we don't use cell lines in my department, but we have a biology and pharmacology department. They do use a lot of cell lines, we can support you. You can send me more detailed information. I can connect you to the specific person and they will help you.
That's great, thank you, Lily. Another question here is, DMPK and exploratory tox assays play an important role in drug discovery and development, that how and when we use them is very important. So what stage should we be using them at?
So all these assays, as you see in the different stage, and it's more specific. Early stage, more you want to quick turnaround. You use in vitro of a few PK studies, and also save resources. For example, we usually use early stage in vitro assays, microsome, hepatocyte stability, and we only do 2 time points and generate some idea and run few PK study looking at the in vitro and in vivo correlation. And then more leader optimization or characterization or candidate, we want to use more more detailed assays. For example, for microsome hepatocytes, you want to generate intrinsic clearance, T-half, CYP inhibition, early stages percentage inhibition, but at this stage we're going to generate IC50s. These studies results, as I mentioned, that they can be part of an FDA submission, and we can put it into the package. All the study we run in ChemPartner, most of the time we don't need to repeat when we go to the IND filing, which is save early virtual companies or small companies lots of resources, in terms of these type of filings, So different stage use different assays, different results to interpret different results. Yeah.
That's great, thank you, Lily. Someone else here has asked if you could please elaborate on the importance of microsome mass shift assays.
This is important. Early stage shifter assay is important because it's very quick. And if you go for the more detailed MAD assays, it takes a longer time. Shifter assays, we can get done 2 or 3 days, get your results and then know potential hotspot. And if you have a detailed, problems, you can send to me, and we are very strong in terms of MAD. We have 14 people's MAD group. They all join backgrounds, organic chemist, and then they will help you. Shift assay is very quick and pinpoint, most of the hotspot for you.
Fantastic, thank you, Lily. And I think we have time for one last question, which is, when do you believe in vitro and in vivo assays are most applicable, and how do you use them?
So again, so for in vitro early stage, you have so many series, like I said, 520 series, different SAR. We can use in vitro to screen early stage. It's parallel with your biochemical and cell-based efficacy assays, more in vitro. Certainly we want, as I mentioned, we want to look at the in vitro and in vivo correlation, sometimes because the compound metabolized by phase 2 enzyme, so you need to use hepatocytes in terms for screening early stage. But with the compound move forward for lead optimization and candidate, we definitely want to use PK. And again, our PK turnaround time very fast. I use the big farmers in US. We do have right now for PK rodent PK study, our turnaround time is about 3 to 4 days. You can get the data right away, but this is more later stage. Certainly early stage, some company wants to use it because in more detail, and also our turnaround time is so fast. They think PK study are good too in the early stage. Again, different stage use different assays, in vitro, in vivo combination. That's all will help your move compound forward.
Absolutely, that's great. Thank you, Lily. I believe that's all the questions that we have for today, which brings the Q&A to a close. So thank you very much to our audience for your questions. And once again, thank you to Lily for this excellent presentation. This webinar is part of the run-up to our Drug Discovery Series 2021, which will be taking place on the 1st and 2nd of June in London, UK. If you would like further information on the Congress or would like a copy of the conference programme, please contact the marketing department or visit the conference website. On behalf of Oxford Global, I'd like to thank everybody for joining us again. I'd like to say a big thank you again to Lily, who has put a lot of time and effort into making sure this webinar was as interesting and valuable as possible. If anyone has any further questions, please do email them across to my colleague using the details on the screen. All that remains to be said is to have a great day, and we look forward to welcoming you to one of our future events. Thank you.